Welcome to seminar. My name is Kyushu. I'm Catalytic Material Transformation Division. I'm an associate professor. Today, I'm very pleasure to introduce Professor Gerard Marriott. He's from the UC-Berkeley in USA. And okay, people, his talk, I briefly introduce about his career. Professor Marriott, he received his undergraduate in biochemistry from Birmingham University, it's a United Kingdom, in 1980. And he got a PhD from University of Illinois. Oh, University of Illinois. University of Illinois, which located your...
There's only one.
Only one? About Champagne?
The other one doesn't matter.
Doesn't matter?
only Urbana matters.
Okay, interesting. Okay, he carried out the postdoctoral research with fellowship from Alexander von Humboldt and JSPS also. Oh, JSPS. In 1982, he moved as a C3 professor to Max Planck Institute for Biochemistry. Oh, biochemistry, okay. He, C3 professor. Was it? Associate professor, okay, I see. And then he worked in the University of Wisconsin-Madison, and then he led a new biophotonic initiative. Okay. And then he moved to UC-Berkeley in 2009, where he researched on the novel biosensor and optical microscopy with the application to high contrast imaging and the control of biomedicals. Okay, welcome. Please start your lecture. Okay, his title is the Application of Optical Switching Prof in Bioengineering and Biomedicine.
Okay, well, thank you very much for the introduction, and thank you very much for the invitation to come here, and also Professor Xu. Thank you for coming here late on a Friday. Very busy time of the year, I know. I appreciate your attendance. And although you can probably guess, my background is very much in the bio field. In fact, I think myself more of a chemist, biochemist, and more recently, material scientist. So I will try to make the lecture of interest to the non-biologists. I think that we have many things in common, as you'll see. Those of you who work with water splitting with light and catalysts, we have a lot in common, I think you'll see. Okay, so this is a picture of Berkeley. It's not the scene I have from my office. I have a view of a parking lot, and I'm on the back side of the campus, so I don't have this beautiful view. Okay, so I'm going to be talking about optical switches. I mean, I think they're very familiar to you from one of the PIs from Urbana-Champaign. We're going to be talking about different types of organic optical switches. This is one of them. It's called a nitrobenzpyran. And here we have a molecule which has essentially two aromatic rings that are divided by that sp3 carbon in the middle. And the two rings are organized orthogonally like this, with the carbon being in the middle. Okay, so the electrons, the pi electrons on this side are not conjugated with the pi electrons on the right side of the molecule. So consequently, this has, this has UV absorption, slightly about 400 nanometers or so. Now, this molecule is under great strain. This bond here, the spiral bond, is very labile. And we'll talk about different ways in which we can break this bond. The easiest way is using UV light. And I should stop this. When you expose this to a short pulse of 365 to 400 nanometer light, this goes to its excited state, and this bond lengthens and ultimately breaks. When this bond breaks, now there's nothing connecting this carbon here to this oxygen, and so what happens is those two rings start to have the freedom to rotate, and they rotate to this planar state here, the meracyne state. Okay, so now the pi electrons are conjugated throughout with this double bond here, bridging the two sides of the molecule. So this is planar. It's a very conjugated system. It's a very polar molecule, and consequently has a very good absorption around about 550 nanometers. So this is largely colorless, and this is brilliantly pink in color. And this reaction occurs with very high quantum yield, and it's complete within nanoseconds. Okay, so we can quantitatively convert this state to this state with a pulse of UV light and very, very quickly. Now, what I was showing you in this animation was that this state here is metastable. The lifetime depends on the environment. In a crystal, it's very stable. It stays in this colored state. But in solution or in many other polymer environments, that planar ring system will go back to the spiro state. This is thermodynamically more stable. Okay, so what happens is we excite this with 365 or 405, we generate this state here, and then this goes back thermally quite slowly, time constant of a couple of hundred seconds, or very, very rapidly if we expose this to green or red light. And that quantum yield is also quite high. So what we can do then, we pulse with UV, we form this state, the meracyne state, and then we excite a few times with 546 nanometer light to reform this state. And then we can do that again and again and again, and we can get a waveform of the conversion of this shape, this profile, but we can change that. We can generate almost any waveform we want, a square wave, sinusoidal, simply by controlling the intensity and the timing between these two pulses of blue light and green light. So what happens is we generate this oscillating concentration of the spiro and meracyne. The quantum yields are high enough that this is very robust. It doesn't really change cycle to cycle. And we can show that this meracyne state here, if we excite it with green light, it goes to its excited state. Most of the molecules, or half of the molecules, will come back here, and the others, or a fraction of the others, will decay back to the same meracyne state and give us a photon, a fluorescent photon. So this is very, very useful, this photon, because we can use it to read out the state of the switch in a sample. For example, if we start off with 100% of this and we excite with green light, since we don't have any meracyne, there's no fluorescence. On the other hand, if we generate 100% of this, we'll get a maximum fluorescence from the meracyne, and as we deplete the meracyne as a result of this reverse photochemical reaction, then the intensity of the meracyne will drop simply because there are fewer molecules. And we can show this, actually this molecule right here is very permeable to living cells, and so if we do this kind of optical excitation, UV vis vis vis, UV vis vis vis, over many cycles, then we can get a inside of the living cell, the oscillation of the fluorescence from this state here. And with this we can do hundreds of times. Okay, so we can control the populations with two colors of light. They're very robust. We can do this many, many times, and we generate defined waveforms of the fluorescence intensity as a consequence. Now that oscillation is generated artificially in our case, but oscillations are very common in biology. But there's almost no way to generate these oscillations of calcium or protein activity artificially, you can only observe what nature does. So the most famous example of oscillation is the cell cycle. Cells divide every 16 hours or so, and the mechanisms that allow them to be so precise in the timing of the cell division related to oscillations are two molecules. There are cells like amoebae that aggregate, and the signaling to form the aggregate results in the diffusion of a signaling molecule called cyclic AMP. And you can see here, these are millions of cells producing molecules, but they do it by an oscillatory fashion. So this is cell-cell communication using oscillatory fields. And then this is a much more exotic example. This is a cuttlefish related to the octopus squid family. And the interesting thing here is that it exploits not only the oscillatory phenomenon, but it does so as a traveling wave. So here's a movie showing the cuttlefish and its traveling wave pattern of coloration. And what this is doing is you can't take your eyes off that, and nor can a little fish that's seeing this cuttlefish. And so the cuttlefish mesmerizes the little prey. It is so interested to see this flashing pattern here, which is a traveling wave of cells becoming dark and light and dark and light. And as it does so, the little fish is looking, it just can't take its eyes away, and the cuttlefish eats it. So it's a very clever way to do to exploit oscillatory signaling, if not somewhat complex. So optical switches have that behavior. We can, generate waveforms of two states very well. And if the properties of those two states differ, for example, if they have a different affinity for calcium, which is a very important molecule in signaling, then we can generate defined oscillations of calcium, which are also very common in the liver, in the pancreas, in insulin secretion. So this is an unexplored area of biology. It requires the integration of chemistry, some photophysics, and some biological engineering to make it work. But reproducing, understanding these mechanisms is very, very challenging without the tools to control the spatial and temporal concentrations of these important signaling molecules and ions like calcium. So just as a quick overview, I'll be talking about these two-state switches in different contexts. In this context, with the oscillation, we've developed a switch, the same nitroBIPS switch molecule, onto which we've appended 4 calcium carboxyl groups. And these carboxyl groups can bind to calcium only in the MC state. The geometry is such that this facilitates the strong binding of calcium, whereas the spiral state, those carboxyl groups are pointing away from each other, so it doesn't bind to calcium. So consequently, if we form the merosanine state with UV light, we get a very tight complex, so the free calcium concentration drops, and then when we go back to the spiral state, the affinity for calcium is much lower, so the calcium is released. So this way, by going to SPMC, we can generate defined concentrations of calcium inside of the cell. And this is one example where we monitor the formation of the calcium-bound merosanine using the fluorescence of the merosanine. And you see the UVs get strong and then weak. Then we hit it again with UV, strong and weak. So we can do this in cells very easily. Here's an intensity profile of this data showing the oscillation. Okay. I'm going to be talking a lot about fluorescence. I think you know the basics. This is just a very simple review. Most organic molecules, the excitation involves transition from the highest occupied molecular orbital to the lowest unoccupied molecular orbital. The excitation is driven to different vibrational levels of the S1 state. And all of the excited states will decay in picoseconds down to the V0 state of S1, from which they will spontaneously decay to various vibrational levels of S0. Okay, so this sets up your excitation spectrum. which of course is of higher energy compared to the emission, because you've lost the energy here from these vibrational relaxations. Okay, so this difference in energy between the maximum of the absorption and the maximum of the emission is the Stokes shift, and this can vary depending on the molecule and the environment. Now, the reason why we're so excited about fluorescence and using it to study molecules inside of living cells and in polymers is that it's very sensitive. We can image a single molecule of a fluorescent dye. Fluorescence emission is wideband from UV to infrared. Fluorescence emission is very fast. The decay is nanoseconds. And as you've just seen in the previous slide, we can use that merocyanine state not only to bind calcium, but also to provide a signal that tells us that calcium is bound. Okay, so fluorescence is used a lot in microscopy, but there are some big problems. And one of the problems is kind of illustrated here. This is a single cell that's actually undergoing what's called endocytosis. This is an active phenomenon where little bits of membrane containing components from the outside of the cell are kind of brought in. It's like a drinking, if you like, and the components from the outside are brought into the cell and finally used to feed the cell. It might be a small molecule or glucose or something like this. So the dimension here, this is about 20 microns in diameter. Each of these vesicles is about 100 nanometers. So we're below the wavelength resolution of the light microscope, but we can see these endosomes, individual endosomes, because they are loaded with a fluorescent dye. Okay, so we're not resolving the actual position because that's below the wavelength of light, diffraction of light, but we can identify their position through the fluorescence emission. Now the problem here is that we're very interested in the components, both on the outside and the inside of these individual endosomes. 100 nanometers, we can calculate the number of molecules. Generally, on the inside, we'll have only about 300 molecules of a fluorescent dye. And on the outside, it's going to be about 10 times less than that, because we're concerned with the surface area. So we have to be able to image just tens of molecules in each of these endosomes. We want to try and figure out what's going on. So imaging very few molecules is very challenging inside of a living cell. The reason primarily is the background. The cell, even without the artificial dye, the artificial probe, has its own fluorescence. And that fluorescence is varying in time and space to generate a background signal. Now the biologists would solve this problem by over-labeling, having a huge number of fluorescent dyes on a structure, but this is not always possible and not always advised, because the dye can influence the cell behavior. The reality, especially shown in the previous slide with individual endosomes, is that the signal from the artificial dye that's on a protein of interest is way below the background. So how can you image this magnitude of signal when the background is 10 or 20 times higher? There are some tricks you can play. I won't go through these, but just the first one shows the problem with conventional microscopy, where the light is focused down onto this image plane here, but of course you have excitation above and below that focal area, and the problem is that if you have molecules of interest, which are shown in yellow here, and background molecules in red, you'll excite both of these above and below the focal plane. So they'll all be imaged in the sample, and so your background is huge. You can improve this by using a so-called total internal reflection excitation, but the events are limited to a few nanometers, tens of nanometers from the surface. And then you can do things like a photoactivator probe, but that's not a reversible process, and I won't talk about that. So the methods that people are using to try to improve contrast are not very good, or not very widely applicable. Okay, so it turns out that this is George Stokes, who in 1842 published his paper in 1852, worked on fluorescence, and he actually was the one who coined the term fluorescence and understood its meaning, as I showed you in the earlier slide with the Stokes shift. And the way that George Stokes imaged with his eye the fluorescence from quinine, which is what you have in tonic water, Canada Dry or something like that. If you put a UV light to a bottle of Canada Dry, you'll see it's brilliantly fluorescent, okay, a beautiful blue-green fluorescence. And that's coming from quinine, which is the tonic in the tonic water. So if you excite quinine with light, in George Stokes' day, he simply took sunlight, put it through a Newton prism, spread the colors, and showed that only in the invisible part of the spectrum did he see the fluorescence from quinine, which he isolated using a wine bottle filter to block out the excitation light. So he showed that UV light, or invisible light in his day, excited the molecules of quinine. They emitted at a longer wavelength that he could enhance its contrast through a glass filter or wine bottle piece of glass, and then he could get very high contrast fluorescence images with his eye of that complex sample. And really, this is the same way we do imaging in the microscope today. Nothing has changed apart from the cost. And okay, the resolution is much higher and the detectors are much better. But the actual way that you isolate the fluorescent signal with a dichroic filter is essentially the same as George Stokes. So there's not much we can do really to improve contrast if we have a system inside of a living cell, as I just showed you, where the probe of interest, here shown in red, is completely saturated by background molecules which have similar fluorescence properties. Okay, so it's almost impossible to resolve the emission from these two with this conventional way of doing imaging, even if you apply higher order separation techniques. So we have developed, or we recognize this problem, and we actually are using optical switches like the one I described earlier as a way to improve the signal to noise of a specific probe in a very dirty environment. And it's actually using lock-in detection of that modulated fluorescence signal that we generate using a two-state optical switch. Okay, so this can be shown here. This is a conventional way of doing microscopy. This is our way. So now if the yellow bordered circles here are the probes of interest and the rest is just background, if these probes have a switchable character like the one I just showed you, then if we excite with two different wavelengths of light, we'll generate an oscillation of the fluorescent signal as we convert one to the other and go back to the original, whereas all those background molecules don't have that behavior. This optical switching behavior is very special. Very few molecules have it, and they have to be synthesized, for example, in our lab. So this behavior is unique to the optical switch. So now we have a spectral discrimination just like George Stokes had, but also we employ what is a simple digital lock-in detection. So we just look for that part of the fluorescent signal that gets through this barrier filter that has that defined waveform of fluorescence signal modulation. And so by doing that, we can isolate the component of the signal that has the oscillation, and if that's done pixel by pixel in a 1 million pixel field of view, then we can resolve regions of the field that have the optical switch probe, and we can remove entirely the background. Just go a little bit over the events again. This is the non-fluorescent state of the probe. We excite with UV light. We go to the excited state. It can either come back down to the original state, we wouldn't see that, or else it can go with the photoisomerization to the methionine state. This is metastable, so we can get reverse reaction here thermally. It's quite slow. Much more quickly, one can convert the methionine to the spiro by exciting with green light. Again, we go to its excited state. It can come back down as fluorescence, or it can go over to the spiro state. So the quantum for this reaction is about 0.5. Every other molecule, excited state molecule, will return to spiro. Okay, so this is the potential energy diagram for this system that I'm going to be talking about today. There are different types of optical switch probe that we've made. In particular, we want to push the action spectrum away from the UV to the visible as much as possible. And so for this, you'll recognize this again as the sp3 carbon of the spiro. So we just append additional pi conjugated groups onto this core in order to shift the absorption spectrum. So we made about 200 compounds in order to identify this one here and its relative here, which are the active ones. They have the strong absorption in the blue to 450 nanometer region, and they undergo robust switching. These are also quite good. They have very good absorption, but they don't switch quite as well. But it took a lot of chemistry to identify this molecule. Okay, so what can you do then to improve image contrast? We take a molecule like this, which is nitrobips with a long lipid tail. Lipid tails embed in membranes, and this is a neuron, or a bunch of neurons, that are taken from a frog egg embryo, from the developing embryo of a frog. And these are placed on the cover slip, and in the frog embryo you have a lot of black material, which turns out to be fantastically autofluorescent. So this is the image of this, the nervous system of the frog, as you would see with your eye or with the camera. So it's not very good. The contrast is very poor, and we have these huge blobs of the autofluorescent yolk from the egg. So this molecule is embedded in the membrane of these cells, and this movie's not working very well, but I'll show you the intensity profile. We get very robust optical switching of this molecule from its spiro state to the myrocyanine state with 365 nanometer light, and then recovery back to the spiro state after exciting with green light, as I showed earlier. So this is the reference waveform that we actually generate by having a bead in the sample, which is coupled with this molecule. So it's like a pure source of the optical switch. And we generate an intensity waveform from that reference bead that looks something like this, okay, over 5 or 6 cycles. So what we do now is we apply this reference waveform to the intensity of for each pixel over the same number of cycles of switching, and from this we generate a correlation coefficient. I think I show later, but if not, I can show you after the talk. And so we simply ask, is there a component in the signal which has an intensity profile similar to the reference waveform that we measured from the pure switch in the bead? And so we generate for each pixel a correlation coefficient, and we can plot that then, this movie with multiple cycles of optical switching. We can operate those images and compute the correlation coefficient, and then plot the correlation coefficient for every single pixel. So now you see here is the cell body of the neuron. These are filopodia. They're very thin extensions of the cell. They're common to the neurons in the frog. And you see that we've been able to resolve these. This is not just a subtraction. This is isolating the oscillating component of the signal. The background is present. It's just not switching. So the correlation coefficient of the background is 0, whereas the correlation coefficient of the lipid probe in very, very strong regions where it's localized can be, and where it's immobilized, can be as high as 1. You see the yolk fluorescence is completely gone because the yolk fluorescence does not switch, so correlation coefficient is 0. So we can improve image contrast simply by looking for this optical switching signal in the fluorescence within a very complicated field like this. So this is what we call optical lock-in detection imaging microscopy. And we can do this in much more complicated ways now, and with much more biologically relevant fluorescent probes. There's a new class of, it's not so new, but proteins that generate their own fluorescence. GFP is the famous example, a green fluorescent protein. And there are variants of GFP that also show this switching behavior. One of them was discovered by Miyawaki-san in Riken Institute some years ago. It's called DROMPA, which I guess is funny in Japanese for some reason. But anyway, DROMPA exists in two states: a trans state where the chromophore has no fluorescence, and a cis state where the chromophore in the DROMPA protein is highly fluorescent. And again, driven by UV and reversed by visible light. Moreover, by exciting with the 488 light, you can also have fluorescence emission. So almost the same as that nitroBIPS molecule. And so we can generate, so the advantage of using DROMPA is that it's a protein and it's encoded by a gene, by the DNA. And so we can take the DNA of the DROMPA and we can fuse it to the DNA of an interesting protein like actin, which is in your muscle, and in the neurons of these cells. And the fusion protein is produced by injecting the neurons with DNA composed of the DNA of DROMPA and the DNA sequence of actin. So the cells will make this protein, and they make it, for example, in this case, in a mouse. So we use a viral, like a virus, to deliver the DNA that encodes this protein. And this virus is designed to go to the neurons only. So the neurons take up the virus, the foreign DNA that we've made, engineered, goes in the cells, and the cells produce the protein. So these neurons are actually in the brain of the animal. They are made fluorescent by the cells in the brain. And this is an image from the brain of the mouse. And this is the intensity image. That's the image the camera sees, so you see with your eye, of the GFP, the DROMPA, the cis state. And this is a real live brain, so you know, it's very dirty in the sense that there's a lot of background. You can see the axons of the neuron. Here's the cell body, here are the axons, here's some other axons from other neurons. But you have problems looking at the connectivity because there are regions where the background actually matches that of the DROMPA fluorescence. So it kind of, you lose the signal, so it looks like this is disconnected. Because this background fluorescence, which arises from natural chromophores inside of the cell and the brain tissue, it doesn't switch, it doesn't have that property. If you use the optical locking detection technique using this analysis of the oscillating fluorescence of DROMPA, and then you compute the correlation coefficient image, and now you see the very well resolved axon from the host cell and from other cells, and all of the interconnections are resolved in the sense that the background has been removed. Rather, you're looking at the oscillating component of the signal, and that's what you're presenting in this correlation coefficient image. Of course, you can take slices. This should be 0.01 to 1. You can take various parts of this range. If you look at only 0 to 0.01, your image would look much more like this. So the correlation coefficient is actually a measure of the persistence of that optical switching in that pixel. And it's also related to the mobility of that one molecule. So let's say, for example, in one pixel, we're starting to oscillate, and then the molecule moves to this pixel. Well, there was correlation for the first half in this pixel, and when the molecule moved here, there's no correlation there, it's just background. Whereas now this new pixel it moved into is now having a higher oscillation in the neuroscience fluorescence, or in this case the DROMPA fluorescence, so it has a higher correlation, a non-zero correlation. So we have two things affecting the correlation coefficient, and the mobility one, or the diffusion one, is actually perhaps the most exciting part of this field, because it tells you about the dynamics of molecules when they are present at levels which are similar to or lower than the background. And there's nothing you can do in these samples. You can't take a brain and somehow clean out all the background fluorescence, otherwise the animal's dead. So this is, you have to live with that background, and you have to resolve it. So we just, borrow the ideas from the electronic engineers with lock-in detection and apply it to this particular system here. The nice thing about fluorescent proteins, you can, as I say, you can encode any cell with them, but you can also find other proteins which switch in different colors, and you can combine two different probes, two different optical switches linked to two different proteins, and you can do dual color imaging. In fact, you can even do 3-color imaging of these optical switch probes linked to 3 different proteins. And the switching in this particular case, this M-cherry Rev protein, is really robust. It's one of the best we've ever seen. Okay, well, any questions while I reset this? So this is optical lock-in detection imaging microscopy. I see I'm running out of time, or I've gone over that. Too long an introduction. So I think I'll skip that part. Maybe I need to show you this. We can take this all the way down to a single molecule by engineering what we call first resonance energy transfer switches. So now we have a fluorescent molecule called TMR that is chemically linked to another switch molecule which is called NISO. So NISO has the property of having an absorption in the far red, which overlaps with the fluorescence emission of the rhodamine probe. So in the spiral state, there's no overlap because this has no visible absorption. However, when we expose this to 365 or 405 nanometer light, then again we break this spiral bond, generate the myrocyanine state, which has a much stronger color in the absorption in the red, and so we get a transfer of the excited state energy from the TMR over to the myrocyanine state of NISO. Consequently, we lose the fluorescence. And so when you do that, you get a remarkably robust on-off switching of the fluorescence of TMR. So this is the spiral state where the fluorescence is really high, and then we can go all the way down to 0 very shortly after converting spiro to myrocyanine as a result of this FRET process. And again, very, very robust over many, many cycles. And we get unique recovery profiles for each of these that we can fit to a time constant. So this can be very fast, and it's actually controlled by the intensities of the light, the 365 and the 546. And so we can, by increasing the intensity, we can increase the rate. So square the intensity, you double the rate. Okay, so the more power you apply at 546 in the visible, the faster this will return to the fluorescent state. And that way we can actually generate kind of quasi square wave waveforms of the fluorescence. And we can do that in a different way by using by imaging single molecules of this molecule, TMR NISO, attached to single actin filaments, from which we can generate the fluorescence state. We can generate a point spread function, the fluorescence intensity in the x and y z plane. The centroid of this Gaussian is the most likely place where we'll find the molecule, and this can be resolved to about 10 nanometers or so. So we can turn this on and off. The movie's not working, but you can see that these fluorescent spots corresponding to single molecules of the OLED FRET sensor go on and off in response to UV and visible excitation. Okay, so we've done that. Okay, so the short exposure of a single gold bar to 639 will heat the gold nanobar. Okay, you get near infrared heating of the gold nanobar it gets very hot, and it provides enough energy, as we'll see, to overcome this barrier here. So how do we know that? So here's the actual experiment. This is a single nanoparticle, gold nanobar, and we're looking at this myrcene fluorescence. So we're exciting the molecule at 550, and we see in the initial state a very weak fluorescence. Some myrcene molecules obviously are stable on the surface. But then we do the 639 excitation of this gold nanobar to heat the gold surface, and we continue to image the myrcene fluorescence, and instantaneously you see the generation of a bright myrcene fluorescence from the same bar. So the myrcene state that's generated that we are imaging here is coming not from 639 nanometers because it doesn't have any absorption. The 639 nanometers is exciting the plasmons in the gold nanobar that are heating the gold nanobar and allowing the spiral state to transition over to the myrcene state. So the myrcene state is generated thermally by optical heating of the gold bar. And the more you excite this with 555, the more you deplete this myrcene state till it goes back to the original condition. And so here we show you can do this gold heating trick to transition between the spiromyrcene states of surface-bound nitrobips over many, many cycles. So we're not going into the excited state here. Everything's done in the ground state. Well, we're overcoming that energy barrier from the spiro to myrcene in the ground state by optical heating of the gold nanobar. Okay, 53. I think. Okay, I'm not going to get to the last bit, but I want to show you optical switches are useful not only for imaging but also for manipulation. And we are very interested in materials that show optomechanical coupling. The reason why we're interested in that is that the stem cells in our body will differentiate into mature cells, and the same stem cell has the potential to differentiate into a neuron, into a muscle cell, or a bone. The same cell. So what differentiates the outcome, the differentiation outcome? One of the most important factors is the surface to which the stem cell is attached. So this is the same stem cell grown on 3 different matrices of different stiffness. This is a kilopascal. This is very, very soft, 1 kilopascal. This is 10, and this is 100. Same stem cell, same condition. The only difference is the stiffness on the substrate to which they are attached. Those on the very soft substrate differentiate into neurons. Those intermediate stiffness, muscle, and those on the hardest tissue we have differentiate into bone. So this is a classic study by Engler et al. That really was one of the most important discoveries in the stem cell field. So the stiffness of the environment is an important determinant of cell fate, and it turns out the cells sense how stiff the environment is because they make attachments to the surface, and they're constantly pulling on the surface and saying, okay, how stiff are you? And this information is relayed to the nucleus to turn on a certain set of genes which will determine if they are brain, muscle, or bone cells. So, very little is known about the mechanosensitivity of cells, and very little is known about how they sense the stiffness of the substrate or the extracellular matrix to which they're attached. The attachment of a cell to a substrate is done through a so-called focal adhesion. So this is the cell side. These are the contractile fibers that the cell produces that are connected to the filaments that are present in the extracellular matrix, collagen. So there's a connectivity between this contractile element here and the polymers in the attached material of the matrix. And so if there's a change in the stiffness of the matrix, for example, as a result of hardening of the arteries, which is very common in the Western world, the stiffness will be sensed by the vascular cells as they tug on the substrate, which has become much harder because of calcium deposits, for example. And that may result in them differentiating into, or cells that are present in that environment differentiating into bone-like cells. You have bone-like cells in your arteries. You know, things are getting worse and worse and worse. Okay. Now, so the cells, most cells, are able to control the stiffness of their environment because there are many components that make the matrix. And so there's a homeostasis is achieved by the cells sensing how stiff it is. If it's too soft, they make more protein that makes the matrix harder. And if it's too hard, they can make proteases that cut some of these collagen proteins to make it softer. So this is called the homeostasis of the biomechanics. But sometimes it really goes awry with in disease states, especially in the arteries of the heart. Okay, so the control of the stiffness of a substrate can be achieved in bulk. You can make a hydrogel or a polymer, a big slab of that, and grow the cells on that. So it has a uniform thickness, and then you take another experiment with a softer slab, and so on. But you can't control the stiffness within a single micron region of a cell, or multiple micron region of a field with cells. Okay, you have to have completely different samples with different stiffnesses. That's the way things work right now. So what we've done is we have taken that NISO switch and we've appended bifunctional reactive probes, labels here, maleimides, for example, and we are able to make hydrogels by cross-linking polymers to these maleimides. It's a little bit like the acrylate chemistry that is used in conventional polymer synthesis. So here's the NISO. It has the two maleimides. We add this to, this is a NISO, we add it to the forearm thiol peg. The peg units are cross-linked by the NISO, so you form a hydrogel where every cross-link is a optical switch. And then we can add other proteins like collagen to make this compatible for cell attachment. This hydrogel goes from spiral to merasanine state the same way as I described. R1 and R2 in this particular case are further away. R1, R2 are closer together in the merasanine state. If we take this hydrogel, looks like a contact lens, and we excite just the left side with the UV, it turns brilliantly blue because of this reaction. There's some spillover here, but you see that most of the blue is over here. And this transition, the same transition I talked about, occurs. And the merasanine state is actually like, in the polymer, it's like a torsional motion. It's like wringing a wet towel. Okay, so you bring about that transition, you move the polymer units in that orthogonal way, and then the merasanine state being metastable, and with the additional tension in the polymer where you've irradiated, you get the reverse reaction. So the blue color goes back to the original thermally, a time constant of about 3 minutes or so. Okay, so here's a blow-up of this gel. This is a spiral state. This is a merasanine, so it's a very robust change in coloration, and the cells will attach to this and grow very well. We can quantify exactly how much merasanine we've made by recording the absorption of these. And then what we can do is we can we'll do rheology measurements where we measure the stiffness in the spiral state and the merasanine state at different energies of the 4 or 5 nanometer light. And we get about a 13% change in the rheology, the macro scale measurement of stiffness for this particular preparation. So 13% is quite decent. It's up on the upper end of that which happens in our tissue. If we take any one of these, we can apply cyclical changes in UV and visible light to control the stiffness. It's a bit noisy. This is a complicated measurement, but nonetheless you see the oscillation of stiffness in response to UV and visible light. How much time do I have?
5 minutes.
Oh, so I thought you were going to say none. Okay. All right, so we have this way of making hydrogels the cells can grow on. We use light to control the spiromeresanine. So we have the ability with the light, in the light microscope to restrict these spiromeresanine transitions to micron regions, subcellular regions. So now we really can investigate the effect of changes in stiffness on cell behavior and even within individual regions of a single cell, because we have the ability to control either on the surface or within a matrix the spiral to meresanine transition in the hydrogel. Moreover, we can make all sorts of waveforms, as I've described, and one of the ones which we're interested in most is the peristaltic motion created by blood flow. So your arteries are expanding and contracting as a result of the peristaltic flow. That is taken up by the muscles around the blood vessels, and of course if the blood vessel hardens, that becomes more difficult. But it's almost impossible to do this unless you do a macroscopic measurement. That is, you have a tube and you flow blood through peristaltically, but you can't look at individual cells this way. So this ability to control with high spatial resolution the stiffness of a hydrogel or polymer that cells are attached to allows us to, for example, look at the effects of a repetitive perturbation, as small as it might be, on the order of 10 or 12% on the cell behavior. There's also this effect here, which is interesting. There's optomechanical couplings. In this region here in the blue is where we excited with 4 or 5 nanometer light, and we would only expect to see the meresanine generated within this region, but in fact, during this experiment, which was a continuous excitation of this box region with 4 or 5, we saw a spreading of the meresanine, showing that we have a coupling beyond this region here. So the formation of the meresanine here it propagates a little bit beyond the irradiated region. Okay, so let's go to this high-resolution study. Here is the boxed region. We excite everywhere here with 4 or 5, and we generate the meresanine, which, because of its absorption, looks black in this transmission image. Within that box, we excite with 555 nanometer light, 5 different boxes, and we generate the spiro state, which has no color, so light transmits through this. So these are cell-sized boxes here that we can generate within a field of view. And within this green region here, we can control the spiro and meresanine transitions that I showed you earlier. So this is just like that experiment we want to do with a peristaltic flow, but we can do this definitely within the dimensions of a single cell, 50 microns or so. And it's very robust, as you see. Okay, so I don't have time to go to details, but some cells hate the stiffer regions. They avoid it like the plague. And there are other cells that like stiff regions. There's actually a effect called durotaxis, where some cells actually like to move towards stiffer regions. One of those is a tumor cell. Tumor cell goes from a very soft region that is created by degrading its matrix to a very stiffer region closer away from the tumor during the tumor spread. Okay, so I'm really getting to the end. We can make more robust changes in stiffness by patterning the hydrogel as an array of microposts. So we have here 2 to 5 micron high posts, 2 micron by 2 micron length and breadth, 5 microns high. So these are made of the NISO hydrogel. We can now go into individual posts and convert them to meresanine and bring them back and control other posts to make them merocyanine. So we have a very smart material here where we can control stiffness with 2 micron resolution. This is made from acrylamide with this acrylate version of NISO. And then okay, so within individual posts we can control, and we can also control the waveform. Okay, so I just want to move on. Here's the image of the posts, a small region, 4x4. We can make the merocyanine, we can go back, we can do this over 12 hours, and the intensity profile of spiromerocyanine is the same for the first 5 transitions as the last 5. So this is 12 hours of data cycling every 10 minutes. Cells love to attach to these bed of nails or bed of posts, and they make very strong contacts, focal contacts with individual posts. This is a movie showing the post. They actually have autofluorescence. This is a cell attached to the post. It's labeled with a red fluorescent dye. And this region here, the yellow region, was irradiated to convert spiro to merocyanine. And so we're watching the cell move, and as soon as we did that irradiation, we made this post, we deformed it actually, so it actually got softer, and the cell really doesn't like that and quickly moves away, whereas the same region can make contacts with other posts that are in normal condition. So this gives us a way to look at stiffness within very small regions of the cell. And I think given the time I should stop here, but I just want to say thank you again for your attention, and if you have any questions, just stop by afterwards. Thank you.